Greetings everyone. I'm Aditya Jaswal, roll number 24 BEC008, a student of National Institute of Technology, Hamirpur. I'm going to give a presentation on UV visible spectroscopy. So let's get started. First of all, let's understand the meaning of UV visual spectroscopy. So, ultraviolet and visible spectroscopy are powerful analytical techniques that provide valuable insights into the structure and composition of molecules by studying their absorption and emission of light. Now, moving further to electromagnetic spectrum and photon energy. So, here, ultraviolet region. The UV region of the electromagnetic spectrum consists of wavelengths shorter than visible light with higher photon energies that can induce electronic transitions in molecules. Visible region. The visible region encompasses the wavelengths that are perceived by the human eye, allowing for the observations of color changes and fluorescence phenomena. From the given figure, we can say that the ultraviolet region of spectroscopy is around 1000 to 200 to around 760 nanometer region. So now let's talk about energy transitions. The absorption and emission of photons in the UV and visible region are directly related to the energy level of the electrons within molecular structures. When UV radiation interacts with a sample, electronic transition occurs. There are four types of transitions: sigma to sigma star, pi to pi star, n to pi star, and to sigma star. We can see the respective transition from the figure. From here, we can say that sigma to sigma star transition needs the highest energy. As we know, sigma bonds are highest stable, and the further required energies for the following transition is n to sigma star, pi to pi star, and then n to pi star. So, where n represents the number of non-bonded electron on a given atom. So the energy required for sigma to sigma star and n to sigma star transition is very high. So that does not fall into the wavelength region of 200 to 800 nanometer as we discussed earlier, which was the region of UV visible spectroscopy. So we do not study these transition in UV visible spectroscopy. So now let's understand the concept of absorption, transmission, and chromophores, etc. First of all, absorption. When a molecule absorbs a photon, its electrons are excited to higher energy levels, leading to characteristic absorption spectra that provide information about the molecular structure. Transmission. The light that is not absorbed by the sample is transmitted, and the intensity of this transmitted light can be measured and used for quantitative analysis. Now further let's talk about chromophores. Chromophores, the word is made up of two words, chromo and phores, in which chromo means color and phore means bearing group. Hence we can say that chromophores is something like color bearing groups or something. So in general, chromophores are unsaturated groups, covalently bonded, responsible for electronic absorption. If we talk about example of chromophores, here in the slide we can see benzene. Normal benzene is colorless, but when nitro group is attached to benzene, it become yellow in color. So what happened here? We can say that nitro group is a chromophore, which impart yellow color to the benzene. So chromophore are further divided into two categories containing pi electrons, containing pi and n electrons. Now let's further talk about oxochrome. An oxochrome is a saturated group, which are not chromophore themselves, but their presence can cause the increase in color intensity of the compound. If we talk about an example of oxochrome, here we can see in the slide OH, NH2, NHR, COH, these all compounds are chromophores. If in details we talk about NH2, NH2 itself is not responsible for color in a compound, but when it is attached with nitro group of nitrobenzene, it increased the color imparting property of nitro group. So what did oxochrome do such that the color imparting property of chromophore increased? Uh, so here comes shifts. Any particular functional group absorbs a particular wavelength in UV visible region, even if they are present in different compounds until their structural environment and solvent is same. If it is changing or absorbing a higher wavelength of light or lesser wavelength of light, then it means something is undergoing in process like absorption shift, intensity shift, or transitional shift is happening. So if we consider the previous example of NH2, that how it increased the yellow color intensity of NO2 group by undergoing some type of shifts. So now let's talk about shifts. So here from the given graph, we can see the various type of shifts. So first one is bathochromic shift, which is also known as red shift. Uh, when the absorption wavelength of light shifts to a longer wavelength, it is known as red shift. And the next is blue shift, which is also known as isochromic shift. So in this, when the absorption wavelength shifts to a shorter wavelength, then this shift occurs. And the next is hyperchromic shift. A hyperchromic shift refers to an increase in the intensity of absorption or emission band. And the last one, the fourth one is hypochromic shift. A hypochromic shift refers to a decrease in the intensity of absorption or emission band. So now further let's talk about Beer-Lambert's law, which states that when a monochromatic light passes through a solution, the decrease in intensity of incident radiation with thickness of solution is directly proportional to the intensity of incident radiation as well as the concentration of solution. So if we see it in mathematical terms, A is equal to epsilon Cl, where A is absorbance, epsilon is molar extinction coefficient, C is concentration, and L is path length. So we can further see the linear relationship. So the Weir-Lambert law describes the linear relationship between the absorbance of sample and the concentration of the absorbing species. Quantitative analysis. This law allows for the quantification of analyte concentrations in a sample based on the measured absorbance at a specific wavelength. Now let's talk about the limitations of Weir-Lambert's law. The Beer-Lambert law has certain limitations, such as the need for a linear response and the potential for interference from the other absorbing species. Now let's talk about the instrument compounds and design. So first of all is light source. UV visible spectrophotometer typically employ deuterium, tungsten, halogen lamps to provide a broad range of wavelength for analysis. Next is monochromator. The monochromator is a critical component that selects the desired wavelength of light to be directed toward the sample. As we can see here the filter which is used in the figure, it is also called monochromator, which changes the simple light into monochromatic light. So now the next is detector. Photodetectors such as photodiodes or photomultiplier tubes measure the intensity of light that passes through the sample. Optical pathways. The instrument design ensures the efficient and precise delivery of light to the sample and the accurate measurements of the transmitted light light. Now let's further see the qualitative and quantitative analysis. So first is qualitative analysis. UV visible spectroscopy can be used to identify and characterize unknown compounds based on their unique absorption or emission spectra. Next is quantitative analysis. By applying the Weir-Lambert law, the concentration of specific analyte in a sample can be determined from the measured absorbance. So next is applications of UV visible spectroscopy. So to distinguish between saturated and unsaturated compounds, to detect the presence of functional group, as we studied earlier, how when NH2 group is attached to the nitro group, it increases the color imparting property of NH2 group and give him a bathochromic shift. So from this, we can detect the functional group. In that case, which was NH2. We can predict extent of conjugation, purity of compound. We can distinguish between geometrical isomers. And the next is applications in chemistry, biology, and material science. Chemical analysis, identification and quantification of organic and inorganic compounds, monitoring of chemical reactions, and quality control in pharmaceutical and industrial applications. Biological application, determination of protein concentration, DNA and RNA quantification, and the study of chromophores in biological system. Material science, characterization of optical properties, band gap determination, and monitoring of thin film deposition and nanoparticle synthesis. Now here let's talk about the limitations and considerations of UV visible spectroscopy. The first one is interference. Presence of other absorbing species in the sample can interfere with the measurement and require careful sample preparation. Sample limitation. Samples must be transparent and free from scattering or turbidity, which can affect the accuracy of the measurements. Sensitivity. The sensitivity of UV visible spectroscopy may be limited for the detection of trace analytes, requiring more sensitive techniques in some cases. Sample volume. The sample volume required for analysis can be a limiting factor, especially for precious or scarce samples. So, in conclusion, UV visible spectroscopy is a powerful analytical tool that opens doors to door understanding molecular structures, concentration measurements, and much more. I hope this presentation has given you a deeper appreciation for its applications. Thank you for your attention, and I would be happy to answer any questions. Thank you.